Okay, so for this lecture we're going to talk about lipids. I had to re-record it because there were some issues on the previous recordings, but doesn't matter. I think the explanation will be a little bit clearer this time since it's the second time that I do it. So anyhow, let me just move. So obviously lipids are a diverse group of molecules that are insoluble in water. You've already seen this in many of your biology classes. Most lipids are hydrophobic, although some molecules can be hydrophilic or have a section of it that is hydrophilic, and we'll see that in a second. So when we talk in terms of nutrition about fats, we are referring to the lipid contents of diets and foods, and so fats is just a group of many different lipid species. And there's basically three categories that we are interested in this course, although there are many variations of lipids that you can find in nature. The ones that are found in our bodies and tissues and in the foods that we eat are triglycerides, phospholipids, and sterols. Okay, and I'll be going over each category, talking about their shape, geometry, composition, and things of the sort. Okay, so the main storage of lipids in the body, basically are triglycerides, and these are analogous to what we've seen, for example, for carbohydrates when we talk about glycogen. So this is the storage form of lipids for most lipids, and in the body we store them as triglycerides. And these triglycerides are non-polar molecules that are composed of four things. So a glycerol backbone, which is a 3-carbon molecule that is bound by 3 fatty acid chains. And these fatty acid chains are composed of carbons, oxygens, and hydrogens. Okay, and so we can classify fatty acids because they are very diverse. We can classify them using 3 things. So you can have different fatty acids based on their chain length. So for example here the carbon chain can be longer, shorter, etc. The saturation level, so when we talk about saturation level we're talking about the number of double bonds between each carbon, and then the shape and geometry. Are they in a cis configuration? Are they in a trans configuration? When we're talking again about the effect that the double bonds have on the shape of the fatty acids. So starting with chain length, there's basically 3 subclasses. Short fatty acids, also known as fatty acids with 6 or less carbons. Medium is between 6 to 12 carbons, and long is between 14 and higher. So unlimited basically. And these carbons, to be able to study their structure, there are 2 poles that are determined in the molecule. So the methyl group at one end is considered the omega group, the omega carbon, and then the carboxyl group on the other end of the fatty acid chain or fatty acid molecule is known as the alpha carbon. Okay, and so when we're talking about double bonds, for example, we'll see that in a second, but we count starting from the omega end. This is why you probably heard of omega-6 and omega-3, right? And this chain length will determine the way the lipid is digested, absorbed, and metabolized. We'll see this at the end of the lecture, but for instance, short fatty acids and medium fatty acids usually don't need any method of transport. They can just diffuse through, pass through the plasma membrane, while long fatty acids require a specific shuttle, which we'll talk about at the end of this lecture. Okay, so the way you name them is usually based on the number of carbons. For example, octanoic fatty acid is also a 8 carbon, octa for 8. Decanoic is 10, and some of them have other names or kind of special names because of their origin. Let's say, for example, palmitic acid, it's 16 carbons, and there's a name for it, most probably hexadecanoic acid, right, for 16 carbons, but they also have other names because they are known to have a benefit, for example, in the for us humans, let's say. Okay, they're found in food sources, for example, like palm oil. So the second concept that we need to look at is the saturation level, and so the saturation level can also help determine or is actually a characteristic of fatty acids, and it's considered as the number of double bonds, or actually when we're talking about a saturated fatty acid, it means that there are no double bonds. So it's actually the presence or lack of double bonds, right? And so saturated fatty acids have hydrogen atoms around every carbon. Now, if they are unsaturated fatty acids, then you will have you won't have hydrogen atoms around each carbon, just because of the double bond, right? And so unsaturated fatty acids have 1 to n number of double bonds. There are the monounsaturated fatty acids, which have only 1 double bond, and then there are the polyunsaturated fatty acids, which have more than 1 double bond. And you can see each fatty acid is actually has a specific function. For example, arachidonic acid, one of its function is to be a pain messenger in your body. Okay, and it has 1, 2, 3, 4 bonds, right? So this concept, I explained it in class, but I think maybe it was not clear enough. So I just want to convey the idea that one of the characteristics that determines the solidity of a fat is its saturation. And the reason for that is saturated fatty acids, they will pack together more easily because they are straight like this. And so you can stack them up one next to the other. Unsaturated fatty acids, because they have these double bonds, the fatty acid carbon chain will bend. And because it bends, it does not allow for the stacking of the fatty acids. And so what happens is that they will not be able to be as solid as something made with fatty acids. Because of that, because they don't take this stacked structure, they end up being a little bit more liquid like this. Okay? When you read textbooks for lipids, for nutrition, online on Google, they make it sound like saturation is the only thing that determines the solidity of a fat. But in reality, it's not, because things like this have proteins in them, they have minerals in them, and so and then the proportion of saturated to unsaturated fatty acids also determines if they're going to be solid like this or not, because nothing is really pure and has only one form of molecule in it, and that's it. This butter, for example, probably contains unsaturated fatty acids as well. The only thing is the saturated fatty acids effect is stronger in this case because of the different species of fatty acids that are found in the butter, because of the different chain lengths that exist, etc. In this case here, probably it's made up of more unsaturated fatty acids than saturated, and probably the saturated fatty acids that are found in it are shorter in chain, because the longer in chain, the stronger the structure it is made when they stack up together. The shorter, if they are short and you have these kinks, it's kind of going to disrupt the solidity. Okay, and so then when we talk about, for example, as I just mentioned, long saturated fatty acids, they do stack well together, and so they form solids at room temperature. If they're a bit shorter, then they have a chance of also making just an oil, for example, instead of something solid like this. Okay? And so monounsaturated and polyunsaturated fatty acids do not stack well together, and because of that, they make liquid fats at room temperature. Okay, but again, not 100% the case for everything, because food is complex. It's not it's only made of this fatty acid and that's it. Usually you have a plethora of fatty acids, and then the proportions of these determine the solidity. Okay, so why are some triglycerides better than other? Well, you can take a look at this graph here, and you can see why do some foods, for example, that are rich in fatty acids are healthier than others. And if you take a look at butter, it's just a question of proportion. They have the least polyunsaturated fatty acids, and butter is known to be not as good for you, for your health. If you took, for example, take a look at walnuts, which are quite healthy for you, they have one of the highest proportion of polyunsaturated to saturated. Okay, and we'll see why this is not quite good for you. Okay, part of it is related to contributing to so your cells, they need to be dynamic, and part of being dynamic is being kind of fluid. The more rigid they are and solid, the less healthy it is for the cell. And so your membranes need to be fluid, and to be fluid they need a certain amount of polyunsaturated fatty acids compared to the saturated fatty acids, because these make rigid membranes and these don't. The second thing, these are more prone to oxidation than polyunsaturated fatty acids, and so oxidation is dangerous, because if you have oxidated lipids in your body, they can trigger a chain of reaction due to the radical electron in them, and that can lead to multiple problems, basically. Okay, destroy other molecules and whatnot. So just a few examples of foods that are rich in saturated, monounsaturated, and polyunsaturated fatty acids. Let's start by saturated fatty acids. So butter and milk, about 65% of the total fat that is found in them is saturated. So that means the rest is either mono or polyunsaturated. So it's quite close, right, as I just mentioned. Coconut and palm kernel oils. So they have 80% of saturated fatty acids. If you read this number, you might think, wow, they're so rich in saturated fatty acids, why aren't they solid? Why is it an oil? Well, as I mentioned earlier, this is the concept I'm trying to pass, is that the oil, the rigidity does not depend only on the amount of saturated fatty acids. Maybe, in this instance, I'm not sure, just an example, maybe all the saturated fatty acids in this oil are short chain, right? So they don't stack as well together as the long chain. And then the 20% that's left of polyunsaturated fatty acids, for example, are long chain. And so these have a bigger effect on the structure of the fat in terms of oil or liquid or solid. Let me see if there's a, what is it? Palm kernel. Is it here? Okay, it's not here. Coconut. Do we have coconut? Okay, I would have liked to see it here to continue explaining about it, but I think you get the concept. Monounsaturated fatty acids, already we're getting a bit in the healthier side of things. As you can see, there's olive oil, salmon, eggs, and so beef, salmon, and eggs, around 50% of their fats are monounsaturated fatty acids. Olive oil is 74%. Cashew nuts, so cashew nuts, obviously, oh no, this is, cashew. I think I forgot to put the 64 here, 60% in the slide. So cashew nuts 60%, olive oil 74%, and then beef, salmon, and eggs 50%. Polyunsaturated fatty acids, already you can see that the food that you are eating is already much healthier than the ones before. So cold water marine fish like tuna, salmon, flaxseed, sea flour, corn oils, walnuts, extremely healthy, and breast milk. And breast milk contains a lot of omega-3 for the development of the kid, and breast milk, omega-3 is polyunsaturated, a polyunsaturated fatty acid. Additionally, this PUFAs, polyunsaturated fatty acids, are essential fatty acids. That means they need to be found in your diet. You don't make them, okay? And so you need to eat them from somewhere. This is why breast milk is rich in omega-3 for the newborn. And the last concept is the shape and geometry. As I said, when you have a saturated fatty acid, it's straight. The molecule's shape and geometry is basically straight. If you are adding double bonds, then these double bonds will start curving, creating these kinks in the shape. If you have one and it's in the cis direction, it will be like this. If it's in the trans direction, it will be like this, etc. And so if you have 2, 3 of them, you can start imagining how it's going to curve the fatty acid. So we're interested in two geometries, the cis configuration and the trans configuration. And the only difference between them is the position of the hydrogen atoms around the double bond. What do I mean by that? When we're talking about a cis polyunsaturated fatty acid, the double bond that is in cis configuration, that means that the two hydrogens are on the same side. Okay? Remember, and that creates this kink here in the unsaturated fatty acid. If we have a trans configuration, then the hydrogens will be opposite of each other, in opposite sides. And this one is straight. Trans fatty acids are straight. Now, obviously, if they're trans polyunsaturated, it might switch a little bit their shape because they're not perfectly straight. But and then you can have obviously a combination of these, right? In the same molecule, you can have cis and trans arrangements. Okay, so you have to think of it as a dynamic thing. Now, you've probably heard from, on TV or on social media and whatnot, wherever you get the information from, that trans fatty acids are not good for your health, and this is because, well, not because, but most of the research right now shows that they are not good for you and that they don't have really something positive for you. And the way trans fatty acids are produced is usually through a process called hydrogenation, which is used by companies when they are generating processed foods. So when you're trying to make some sort of cake or whatnot, and you want to make the fatty acids in it more stable and durable, so that your processed food can, for example, survive for a longer time and resist oxidation and chemical decomposition, then they use a hydrogenation process, which all it does is that it takes unsaturated fatty acids and turn them into saturated fatty acids. And that's by adding hydrogen atoms. And so this reaction, what it requires is an H2 gas, hydrogen gas, heat, and a metal catalyst, for example, nickel. Okay. Saturated fatty acids are will increase your cardiovascular disease chances because they act in a similar way to saturated fatty acids, right? They are more prone to accumulate in plaque, for example, and they will increase the level of bad cholesterol and decrease the level of good cholesterol. And we'll see this at the end of this slide. What do I mean by that? Okay. Now, if you're trying to choose for a diet what food to eat, you're going to kind of try to have more polyunsaturated fatty acids than saturated fatty acids or trans unsaturated fatty acids. The thing is, it's not that easy to see, right? Because if you're buying a cake, it's not butter where okay, this one has 65% of fatty acids, saturated fatty acids. It's not butter where you can be like, yeah, it's solid, so it must be more saturated than unsaturated, and so I will not eat too much of it. In reality, these trans fatty acids are kind of invisible because they are in foods that are processed, that look good, taste good, and so you don't know that they are there. For example, if you buy a donut, it's probably full of trans fatty acids, but you don't know that because you don't see it as oil or as a block of butter, right? And so this is the danger about them. This is why Health Canada obligates companies to declare the amount of trans fatty acids in their foods and to actually put it in front of the thing. Where, you know, sometimes you go buy something and it says no trans fatty acids. Well, or actually it doesn't say fatty acids. It says no trans fat. That's what it means really. And so a complete hydrogenation will remove all the double bonds in the molecule. A side effect of chemical hydrogenation, when it's not complete, you end up with a trans configuration. Okay. Another concept that is important for fatty acids and for oils is the smoke point. So the smoke point is the temperature at which oils and fats start to break down. And this is a point where you don't want really to reach, because that will decompose the fats and lead to their oxidation. And as I said, oxidation is dangerous because it creates these molecules that are toxic for you. Okay, so you don't need to know these numbers. It's just to give you an idea. Olive oil, for example, has 190 degrees of smoke point, while sunflower oil has 246 degrees Celsius. Okay, good. So I just talked about essential fatty acids. So when we're talking about essential fatty acids, it means, in other words, that they need to come from the food, because they cannot be made in the body. One example of this is linoleic acid, aka omega-6, and also alpha linoleic acid, omega-3. So these are important for phospholipids in red blood cells, neurons, myocardiac cells, because they will form, they are first polyunsaturated fatty acids, right? And second, they are very important for the structure of cells. For example, in neurons, you know how you have these myelin sheaths around them? Well, these myelin sheaths, for example, are rich in these lipids, in these two essential fatty acids, and having a disbalance in their levels can lead to actually action potentials not happening. Okay, so they're very important for the brain. Same thing for red blood cells. You need the right mixture of fatty acids and lipids in your cells so that the cells can function properly. Okay, and I'll show you that in a second. So obviously fat intake deficiency is rare. This is one of the things where we just eat. We don't really, because it tastes good, right? Fats usually taste good, and so their deficiency is quite rare. The main deficiency is usually in the essential fatty acids found in them, in certain fatty acids. For example, eicosapentaenoic acid, which is also a 20 carbon molecule, and docosahexaenoic acid, which is a 22 carbon molecule. Okay. So let's look at a little bit structures. The way we write, because fatty acids are so diverse, we have to use some sort of notation, some way of writing it to know which one we're talking about, right? And so for linoleic acid, for example, we'll be writing it like this. So the first number you see in the series of 3 numbers is the number of carbons. The second number is the number of double bonds found in the molecule. And the third one is the position of the first double bond. And so if we're talking about an 18 carbon molecule, so this is it, you can count the carbons, 2 double bonds, here's one here, and then you have omega-6. Omega-6 means we will count 6 carbons from the omega end, which is the methyl group, until we reach the 6th carbon. This is where the first double bond is. Okay, if we take a look at alpha linoleic acid, it's pretty much the same thing. We have 18 carbons, 3 double bonds, 1, 2, and 3. And then the first double bond is at the omega-3 fatty acid. So 1, 2, 3. Do not count from the alpha end. Okay, it's not because the name here is alpha linoleic acid that means you're going to start counting from here. It has no link whatsoever. We always count from the omega end. Okay. So where are these two fatty acids, these two essential fatty acids that I talked about previously are found? So eicosapentaenoic acid, or EPA, and docosahexaenoic acid, DHA, are found in fish and fish oil, and they are very important for reducing the risk of heart disease. They will stimulate prostaglandins and thromboxane to reduce inflammatory response, and to prevent blood clotting in your body. So thromboxane will reduce the blood clotting, and prostaglandins will reduce inflammation. And usually when we talk about inflammation, most of the time it's not good. Okay, inflammation is quite dangerous because it can lead to many problems, one of them being cancer, another one is heart disease. Okay, and so yes, the omega-3. So the omega-3 eggs, I'm remember in class I asked you guys and you said that yes, you've seen these. So I've always wondered how they made this, right? Were they modifying the cells directly that with a gene that would produce more omega-3? Well, actually the way they do this is that they feed the chicken or the hens 10% flaxseed diet, and then these eggs end up having 10 times more omega-3 than regular eggs, to kind of increase the amount of omega-3 in these eggs. Okay, so this is quite interesting. There are many people that are working to increase omega-3 in many things. For example, one of the company that I worked with, they were genetically engineering microalgae to produce higher levels of omega-3 to then use it as part of fish feed aquaculture, so that the fish themselves end up with higher amounts of omega-3. Okay. Now, one thing that I need to tell you about omega-3, actually I think I have it. I have a slide about this at the end. So let's move to the next category, the phospholipids. So phospholipids have a somewhat different composition compared to the triacylglycerols. Phospholipids have a glycerol backbone. They have the two fatty acid chains. So this is somewhat similar to triacylglycerides, but in addition they have a phosphate group. Now this phosphate group is usually bound to an amino acid like serine, which would make phosphatidyl serine, or a choline, which would make phosphatidyl choline. And these are just different types of phospholipids. The two fatty acid chains are the hydrophobic tail. The phosphate group bound to an amino acid is usually bound to an amino acid is a hydrophilic head. And so these are quite fit to produce the plasma membrane of the cell, which is basically separating two environments that are hydrophilic, right? Because they have a lot of water in them. And so phospholipids' main function is to basically serve as membrane for different components of the cell, for example, mitochondria and whatnot, or for the cell membrane itself. Okay, and they're very important for digestion of dietary fat, and we'll take a look at this in a little bit. And so here's the structure. We have the glycerol backbone, so this is nothing new. We've seen this before. You have the fatty acid chains. There's two of them only, because the third carbon instead is bound to a phosphate group and some sort of amino acid. Okay, or other molecule here. And this is the hydrophilic side, this is the hydrophobic side. Okay, and together they will form the bilayer, where you have water on both sides. Well, not really water, there are other molecules and whatnot, but highly concentrated in water, that's what I mean. So you'll have a separation like this. So actually, if you take phospholipids and mix them in a tube and then let them sit, they will form this kind of shape, because they will aggregate together. Okay, so in here, this environment here, all the yellow here is hydrophobic, and heads are hydrophilic, and this gives them the opportunity to be soluble in water. Okay, now the membrane is actually quite complex. It's one of the most complex parts of your cells, and it's highly dynamic in the sense that it keeps changing, moving, and whatnot, and it's bound by pretty much any type of molecule you can imagine. You have carbohydrates, you have proteins, like for example the proteins of the extracellular membrane, you have transmembrane proteins which are passing through the hydrophobic part and through the hydrophilic part. So this is interesting. This means that this portion here of the protein needs to be able to interact with the hydrophobic section of the membrane, while this portion here needs to be hydrophilic and interact with the hydrophilic part of the lipid. You also have the membrane interacting with cholesterol, so cholesterol will come and lodge itself inside the hydrophobic portion of the membrane, and this is dangerous because cholesterol increases the rigidity of the membrane, and if the membrane is rigid, you'll have issues because the cells, the proteins will not be able to take the proper configuration, and the membrane will not have the proper curvature, and that's not good for the cell. Okay. Additionally, lipids can bind to carbohydrates, as I said, and so the hydrophilic part of the phospholipids will be bound to these. Okay. So as you can see, there are different shapes, different formats, different needs, and so you need a variety of side chains. You need a variety of phospholipid heads, and so this is why we have many different combinations possible. Okay. For example, here, let's take a look. For example, here you see this curvature here. This curvature here would not happen if you had only saturated fats here. You need unsaturated fats that will create the curve of the membrane. Okay, this portion of the protein is quite straight, and so you need most probably saturated fats here to interact with the full length of the protein. This protein here, for example, is kind of, or maybe, obviously the diagram is not fully, just to explain the concept. Here it's kind of curved. Because it's curved, you probably need the polyunsaturated fatty acid to interact with the protein here. Now obviously this is a diagram, quite ugly, so it's not very representative of what's happening. Okay. Sterols, these ones you probably know them. You have been watching them, maybe not. So sterols is what make cholesterol. It's just basically lipid molecules that have multiple rings in them. So the other two we've seen don't have rings. These ones have rings, and it is made in our body, so it is not necessary in our diet. This is very important. We have a full pathway that produces cholesterol called the mevalonate pathway, and it's important that it is made in our body because many hormones are based on it. Okay, for example, estrogen, progesterone, testosterone, dihydroxytestosterone, or the sex hormones are made from it. Adrenal hormones, for example, epinephrine, cortisol, and whatnot, vitamin D, and the bile salts that I told you about. So, yeah, I don't know why this slide is here. Who cares? I think I flipped them. And so, cholesterol is a steroid. Okay, it's the component of cell membrane, but also serves to produce, as I told you, the hormones and the vitamins and whatnot. And another function of cholesterol is to act as a precursor to produce bile salts, which are important for digestion of the lipids. If you remember, I told you that bile salts need to emulsify the fat droplet that comes directly in the when we eat food, or that is produced in the stomach when we eat food, while bile salts are important because they will come and break that major structure into smaller droplets. Okay? And so the bile salts are just cholesterol derivatives that are mixed with sodium, okay, that are bound by sodium. Cholesterol is the most occurring sterol in the diet, and its storage form in food is known as a cholesterol ester, and so it's basically a cholesterol group with a fatty acid bound to it. Okay, and this is just your general structure of the sterol molecule. Obviously, they will have extensions and other molecules bound to them, and different double bonds and whatnot. This is the basic structure. If you modify it, you'll get cholesterol like this. If you modify it some more, you can have other different sterols. Good. So that is the part about structure. Now for the digestion of lipids, we've seen this in general, but let me just review it. So the way digestion of lipid happens is that it starts in the mouth, okay, but only to a very low amount. And you have this lingual lipase that is found in your mouth. It will start digesting triacylglycerides, and only triacylglycerides, okay? But it's like 0.01% of the total fat. Why? Because the fat you eat takes time to digest, to break down, and because it takes time to break down, it does not have the time to be broken down in your mouth. Not like carbohydrates, for example, which are easily oxidized, okay, or broken down. And so the first part happens in the mouth, second part happens in your stomach with the gastric lipase. And so the gastric lipase will be breaking a little bit the lipids in your stomach, mostly the triacylglycerides, but the major digestion and absorption of lipids happens in your intestine. And the way this happens is that when the food reaches your intestine, bile will be secreted into from the gallbladder into the intestine. So it's produced in the liver, stored in the gallbladder, released in the intestine, and it is released upon the secretion of two molecules, cholecystokinin and secretin. These will just tell the gallbladder to contract and release the bile. And the only role of the bile is to break down the fat that you ate and that was a little bit processed in your stomach and in your mouth into smaller droplets known as micelles. So these micelles are a single layer of phospholipids with a hydrophobic core and a hydrophilic membrane, not bilayer, monolayer. And these micelles, all they will do is that they will capture pieces of lipids and then carry them where they have to go. Okay? So in your intestine, you also have these pancreatic juices that will be released in it from the pancreas. And these pancreatic juices contain enzymes that are lipases that will break down triacylglycerides into diacylglycerides and monoacylglycerides. Okay? So this is the process of lipolysis. Additionally, when you're dealing with cholesterol esters, you need a different enzyme, and this different enzyme is cholesterol esterase, which also will be released in the intestine to break down the ester bond between the cholesterol molecule and its attached fatty acid. Now for phospholipids, which come from food, you need another enzyme, and this is the phospholipase. And there's many different variations, and they each will break part of the phospholipid. Most of the time, it's removing the head from the fatty acid chain. Okay? So let me see. Did I say everything here? I think I talked about this. Gallbladder, phospholipids. Yeah, I think that's it. Okay. So when you break down the lipids into their smaller components, then you need to absorb them. And so the micelle is the transporting molecule, the initial transporting molecule, and there are other molecules that will transport the lipids. The initial one is the micelle. The micelle starts from the intestine to the enterocyte, which is the cell, the absorbing cells found on the villi of the intestine. Okay? And the micelle, as I told you, comes from the emulsification of the big fat droplet that is made in your stomach, and so it's made up of bile salts and a monolayer of phospholipids. Okay, so it looks kind of like a circle, let's say, or more like in 3D it's kind of a shuttle, right? It has a spherical shape, and the micelle will capture any component it passes by. So remember, we just digested the bigger lipids, like the cholesterol esters, triacylglycerides, and whatnot. And so now we have them in forms of free fatty acids that are just floating, free cholesterol, maybe some diacylglycerides, maybe some monoacylglycerides, maybe some just glycerol molecules floating by themselves. And so the micelles will capture them because they have a hydrophobic core, and so they will just take them as they pass through the intestine and then transport them to the enterocyte for absorption. This here is just showing the breakdown of the triacylglyceride. Okay, I think this slide should have been a bit earlier, but basically you will remove one bond here, or whatever, one here, and you'll leave a monoacylglyceride. So this is the emulsification of the big fat droplet that comes from food, because the bile salts will bind to it. And the way bile salts work, because they are cholesterol molecules, in part cholesterol molecules, is that, as I told you, cholesterol molecules will make the membrane more rigid, and so it will break into smaller pieces like this. Okay? And then the lipases will come and break down whatever is found in them. So the micelle looks like this: monolayer, hydrophobic core. It will take anything that it captures, which are the components of lipids, inside the enterocyte, and then inside the enterocyte, now we have to rebuild the more complex molecules. So we take fat that is complex, we break it into components, we bring it inside the cell. Now we will rebuild those fats to carry them to the cells. Okay? And in the enterocyte, we will remake the triacylglycerides that we just broken down, and we will package them into lipoproteins and release them into the bloodstream, but not directly into the bloodstream. Okay? First, they will go to the lacteal, which I talked about in lecture 2, and then from the lacteal, they will go to the bloodstream. Good. So micelle inside the enterocyte. Now from the enterocyte, what's the shuttle called to take the lipids to the lacteal? And so these are called lipoproteins, these shuttles that I'm going to talk about, and there's 4 of them that we are interested in this course, and they are basically a lipoprotein. So they're composed of proteins and lipids. The first one that we'll talk about is a kilomicron. So the kilomicron is produced in the enterocyte and will transport lipids from a meal, so dietary lipids, to other parts of the body. Okay? So the kilomicron is composed of fatty acids. Inside of it, you have the phospholipids and proteins that I just told you about. They are soluble in water, and they are going to travel through the lymphatic system and then transferred to the bloodstream. In terms of short and medium, so these kilomicrons, they will carry the triacylglycerides, for example, the phospholipids, cholesterol esters, and whatnot. Okay. Short and medium chain fatty acids are just absorbed easily. They don't need to be arranged into the kilomicron, and so they will be just transferred directly. Okay. This is what a kilomicron looks like, or just a lipoprotein in general. You have a monolayer of phospholipids. You have proteins, and these proteins will serve to kind of direct the kilomicron to the specific place that they need to go. You have inside of them is a hydrophobic core, which contains triacylglycerides, cholesterol, and whatnot, and outside is the hydrophilic side. Okay, and they serve as shuttles to carry these. Again, let me just show you this. So I said micelle. The micelle contains broken down pieces of lipids that were digested in the gut lumen. They will go inside the enterocyte. The enterocyte will release their content. The short and medium fatty acid chains will go directly to the bloodstream, while the components of the other more complex lipids, for example, phospholipids, cholesterols, fatty acids, monoglycerides, and glycerols, will be packaged into a kilomicron and then carried to the lacteal. Okay. Now, in the enterocyte, something happens additionally to that, is the rebuilding of the triacylglycerides from its components. Okay, so it's here, triacylglycerides are broken down into pieces. They come inside, they are rebuilt into the endoplasmic reticulum and packaged into the kilomicron. Okay, and the endoplasmic reticulum will generate the kilomicron and package it. Okay. In terms of structure, this is what a villus looks like. We've talked about this in your intestine, and so it has the bloodstream in parallel with the lymphatic system, and so the short and medium fatty acids will directly be absorbed into the bloodstream. The kilomicrons, which contain tons of lipids, are going to go through the lacteal, which then will bring them to the bloodstream. Okay. Ooh, a lot of information. Next. So, fat is used for energy, and it's a long-term storage of energy. So it is stored in your adipose tissue, and your adipose tissue is found at specific places in your body. For example, on your belly, right? And so it's used sometimes when you have energy needs, direct energy needs, it will be used for that. Sometimes it will build lipid-containing compounds that are important for cell function, but most of it will get stored in your muscles and adipose tissue for later use, right? There's an enzyme that will break down the lipids, the triacylglycerides that are found in kilomicrons, and it's called lipoprotein lipase. And this is just again to do the same thing that was done in the gut lumen. So you want to break the triacylglycerides so that they can pass through the membrane and enter body cells. Okay, so you get them from the food. They are broken down in your gut lumen into their components. They enter the enterocyte as a micelle. They are rebuilt into triacylglycerides, packaged in kilomicrons, and then brought to other cells where they are disassembled by this lipoprotein lipase before they enter the cell. So inside the kilomicron is this enzyme here that will break them down. Okay, I know it's kind of repetitive, but this is how we evolved. Kind of weird, but if you think about it, you would want, why not just, okay, we're eating those lipids, we'll keep them as is and then just use them. Well, actually, the reason why we're breaking things down and rebuilding them is that so the body controls the amount it's producing of each lipid, right? You want to make lipids based on your needs, not based on what you eat, because otherwise we'd be in trouble. Because we clearly cannot follow a proper proportional intake of the different lipid classes. And so our body needs to take care of this by breaking down things, transporting them, rebuilding them. So how much fat do you eat? So the AMDR for this is 20 to 35%. And as I said, this depends on which phase of your life you're in and what type of lifestyle you're going through. And so usually you should try to stick to this. So 20 to 35% of the calories that you take should be fat. Okay. You should minimize saturated and trans fatty acids because they increase the risk of heart disease due to their ability to get packaged more tightly and in more rigid forms. And this is dangerous for heart disease because they can block arteries, for example, and lead to a heart attack or a stroke. If we look deeper into the specific lipids, some of them we have numbers for them of how much you need to take. So for example, essential fatty acids like linoleic acid, alpha linoleic acid, we know how much we need of each of them. So linoleic acid, aka omega-6, you need 5% to 10% of your total energy intake. This is a lot, okay? Because the category for lipids, all fats, you need 20 to 35% of your total calories. And I just told you that omega-6, you need 5 to 10%. So this is quite a big number. Omega-3, you need 0.6 to 1.2% of your total energy. So the AMDR is 0.6 to 1.2% of your total energy. You've probably seen that omega-3 is good for you, and so you've probably bought supplements to increase the levels of omega-3 in your body. In reality, it's not just the amount of omega-3 that's sufficient. You need a ratio between omega-6 and omega-3, and this is the ratio that should be followed. Okay? So it's quite hard to do because you don't know how much of these is found in your food, how much of these in your body you have. Usually, the pills that you buy for supplementation of omega-3 have some numbers on them, and they tell you eat one pill a day or whatever. But in reality, those pills are not based on clinical trials. Okay, to get something approved for Health Canada for supplements, you don't even need to do research on it. Okay, you guys right now can start selling supplements as long as you support the claims with some scientific papers in mouse or whatnot. It's sufficient to sell supplements. And so those numbers are not precise. You could be taking more than this ratio. Okay, so we usually see omega-6 as being bad and 3 as being good. Well, in reality, no. It's just the ratio of them that is important. So that's for these essential fatty acids. Saturated fat, try to get less than 10% of your total energy, and trans fats to an absolute zero. Reduce them to an absolute zero. Research has shown in Canada that our omega-3 levels are quite low. So as I told you, different places in the world have different accessibility to foods, and so they will have different deficiencies, let's say, right? For example, in my country, beef is quite expensive, so red meat in general is quite expensive. So in my country, people usually tend to go to white meat, or including chicken or fish, because it's the cheaper form of, the more accessible form of fish, right? And so because of that, we don't have much of the omega-3 deficiency, right? But in my country, we have another issue, which is anemia. So anemia is quite prevalent in my country, and that's due to the lack of eating foods that are rich in iron. In Canada, because we eat too much beef and we eat too much chicken, we kind of lack, we don't eat much fish. Maybe you do from once in a while, maybe once a week or whatnot, but most of the diet is composed of, at least this is what research shows, is composed of beef, is composed of chicken, sometimes salmon, and then, you know, but you don't eat fish every day like you do beef or chicken, right? And so Canadian diets are known to be low in omega-3, right? And so people need to be careful with this and eat more of this. And so most people tend to supplement in omega-3, okay? There are things that you can do to improve your nutrition. One of them is to switch for healthier fats without increasing the total intake. For example, because olive oil or canola oil have a higher percentage of polyunsaturated fatty acids, so then you could use those to replace butter or margarine, right? And then you can also select low-fat or non-fat dairy products. So I know that most people buy 2% milk, you know, from Kibon or Lactancia or whatever. Well, there is also the 1% and skim option. Skim milk is just a milk without any fat in it. Okay. So omega-3 rich food. So if you take a look at salmon oil, for example, it is the richest in terms of omega-3, and then you can go down. So you can see here which foods have high amount of omega-3. And then for milk and whatnot, you can take a look at the amount of fat found in them. So the 3% has 8.4 gram of fat, while the skim milk has 0.2 gram of fat. Okay. Now obviously, removing fat completely, that's another story. This is a problem also. So it's a question of having healthier fat molecules than the unhealthier ones. Okay. So this is why you have to follow this. This is why stopping not eating fat food at all is kind of not good for you, and people try to do this. In reality, it's not completely removing the problem, but switching the subcomponents of it, right? So for example, the ketogenic diet, you remove carbohydrates completely. Well, that's not good. It's not the solution. You need carbohydrates. Some of them are necessary for your body, right? So it's the same thing with the lipids. Why do we need lipids? Well, many things. So the major fuel source for your body between meals is not carbohydrates, it's lipids. You need them during sleep. While you're sleeping, you're burning lipids. I guess not that much, but it's a good way of losing weight, I guess. During fasting, so fasting is quite interesting. It has a lot of health benefits. You know, either the voluntary or involuntary form of it. Sports players, they like fasting, and usually they will be fasting before going to, for example, get their weight before a, let's say, UFC match or whatnot. They'll be in a fasting state. So lipids are the ones that are getting broken down during the fasting period, and they use it to kind of decide which category of weight they want to compete in, right? During physical activity, if you remember the slide that I showed you with the person walking fast, biking, or swimming, and so low to moderate energy exercise that is for extended periods will burn fats more than glucose or sugars. Okay, they're also important. Lipids are also important for the membranes. They are constituents of cell membranes. They fight infections. Certain lipids will serve as toxic molecules to kill some sort of to kill bacteria and whatnot. There's a concept where called there's a self-death form called liponecrosis, and this liponecrosis is death of a cell caused by lipids. And so there are certain lipids that if they accumulate in your cells, they can kill you or kill the cell. Sorry. So these are used to fight infection. Lipids also serve as signaling molecules to tell your body to start fighting these infections. And as I told you, the point of entry of viruses or in your cells is through these lipids, and so they're very important, carbohydrates and lipids, and so it's very important that your cells have the right lipids in their membranes to avoid getting infected, for example. Okay, and one of the most interesting concepts is that lipids are important for growth. This is the blue whale. It's the biggest animal that exists, and so the blue whale's calf, so that's their baby, grows. So the mother kind of throws this blob of fat in the water, and then the baby just absorbs it, and the calf can grow up to 90 kilograms per day. So this is kind of visible to the eye. If you stand there for a few hours, you'll see that the calf kind of is growing during the same day, right? And so this is quite cool if you get a chance to see this in life. But this is how important lipids are. So this blob that the mother kind of throws in the water is very rich in lipids. Okay. Additionally, lipids are important for the brain, for the visual centers, because they form these sheaths around the neurons. In part, there are other functions, of course, these myelin sheaths, and so this is one of the functions. What else? They also serve as fat-soluble molecule. They transport fat-soluble vitamins A, D, E, and K, and so these lipids, they're very important because they will help the transport of these vitamins to the right places of the body. So these vitamins also are absorbed as part of the micelles, and they're transported also in the kilomicron that I just talked about. Okay. What else? I mean, this is everything that you know that I just talked about. So I think you could just read them. Lipids are also very important for insulation, to regulate body temperature, let's say. So the more fat you have, the less prone to freezing you are, let's say. The others I talked about. Yeah. So why is it important to regulate the amount of lipids in your body? Well, the main reason is that you want to prevent cardiovascular disease. And cardiovascular disease can happen through many problems, for example, a valve problem and whatnot, but usually it is due to a mechanical problem. For example, the valve not closing properly, the artery being blocked, some defect in your heart. Okay, and that could lead to your heart having to work harder and lead to a heart attack or a stroke. The difference between a heart attack and a stroke is that the heart attack is due to a vein that is not in the brain, while the stroke is due to a vein or artery that is in the brain. Okay. So let's see. So there are a few cardiovascular risk factors for cardiovascular disease, and these can be being overweight, so this is due to fat. Physical inactivity also leads to accumulation of fat. Smoking is due to the fact that smoking affects the level of cholesterol in your body. Okay. High blood pressure, so this is again probably due to fat in your arteries. Diabetes, which leads to also, which can be caused by fat, mostly sugar, right? And so diabetes can cause problems with your heart as well. So what's the link between fats and cardiovascular disease? Well, fats will increase the blood lipids level. And when you have too much blood lipids, you can cause a few problems, like block your arteries, right? And this is not good. If you have highly saturated and trans fat intakes, so you eat too much of these can increase the levels of blood cholesterol and lead to rigidity in your membranes, also accumulation as plaque in your arteries, and that can clog your arteries. And so one thing that omega-3 are also good for is that they reduce blood cholesterol. Okay, so obviously supplementing with omega-3 can be helpful to reduce blood cholesterol. The major concept that I want to talk about, the main reason of heart attacks is usually plaque. Okay, and when you have a healthy artery like this, and then you eat too much fat, what happens is that your blood will contain a lot of fat, because the lactate will be just throwing these lipoproteins in them. The short and medium fatty acids will be already in the bloodstream. So when that happens, what happens is that they will get oxidized by other molecules that are found in the bloodstream. And so when they get oxidized, as these lipids get oxidized, this is dangerous. It will create an inflammation response, which will bring these cells to the artery, these immune cells, which will try to eat up the oxidized cholesterol or fatty acid or whatnot. When they eat them, it will transform these cells into what's called a foam cell. So the immune cell will become a foam cell, and these foam cells are quite sticky, and they will stick to the membrane of the artery and start forming this space where lipids will accumulate. So the more you eat, the more they accumulate, and they will form this plaque of lipids here. And this is kind of a cycle where you have foam cells eating, immune cells eating these oxidized lipids, transforming into foam cells, sticking here, more inflammation, more immune cells, more foam cells sticking here, etc. And that will decrease the diameter of the artery, and by decreasing the amount of space that your blood has to flow, this will put a strain on your heart and can lead to a stroke. Okay, so I told you that there are these shuttles that carry lipids in your body, called lipoproteins. The first one we talked about is kilomicrons, but there are 3 others that we are interested in: the very low density lipoproteins, the low density lipoproteins, and the high density lipoproteins. And they all serve a specific role. In this case these are just the compositions of each one of them. I don't need you to remember this by heart, but it's just to show you that as this shuttle travels through the body and is transformed from kilomicron to VLDL to LDL and whatnot, their composition will change. And their composition is representative of what happens to them. For example, if you take a look at the kilomicron, they have a lot of triacylglycerides, right? And so as they travel to the body, they will lose the amount of triacylglycerides because they will get digested by the lipoprotein lipase. Remember I told you about the lipoprotein lipase a few slides ago? Well, this is it. So the kilomicron is made in the gut, specifically in the enterocyte. It will enter the lymph through the lacteal, and that will be then taken to the blood. They are the largest lipoprotein with the lowest density, okay? And they will be taken up by the liver once these triacylglycerides are removed. Then in the liver, the kilomicron remnants, so the remaining portion of the kilomicron with less triacylglycerides, will form the VLDL. And this happens, part of it in the intestine because of the triacylglycerides that get metabolized in the intestine already, but the major part of it is in the liver. And these will travel from the liver to other parts of, like for example, the muscle cells and the adipose tissue. Same thing with the kilomicrons. They will go to the liver, but they will also go to the muscle and to the adipose tissue. The VLDL, as it's traveling, the triacylglycerides will also be metabolized, that are found in them. And by being metabolized, they will become something called a low-density lipoprotein. The low-density lipoprotein is very rich in cholesterol. Because it is rich in cholesterol, that means not good for you. And so the LDLs are not healthy, okay? Their levels need to stay low in your body. And so your body has a mechanism of getting rid of them through the HDLs. The HDLs are also known as the cleanup crew. So as you're going through digestion, your LDLs will be mostly rich in cholesterol, and so the HDLs will come after them and pick up all the bad cholesterol and bring it back to the liver so that it can take care of it. Okay? One important thing to note, kilomicron will carry lipids from your diet. VLDL will carry lipids from your body, so endogenous lipids, because this is formed in the liver mostly. Kilomicron is formed in the gut, so it carries lipids from food, lipids from the body that are found in your liver and whatnot, other molecule, and this is the cleanup crew. If you want to remember which one is the healthy and which one is the bad one, you can use this trick. So HDL, healthy cholesterol, needed in high amounts. LDL, lousy cholesterol, needed in low amounts. Okay. This figure here, I just talked about it. It's where the kilomicron comes from your intestine, goes to muscle, adipose, and the way there's some fatty acids and glycerol molecules that are being kind of released from it and broken down. Whatever is left is called the kilomicron remnant, which doesn't have triacylglycerides anymore. It goes in the lipids. It gets filled up in the liver. It gets filled up with triacylglycerides that are found in the liver, so endogenous ones, to form something called the VLDL. The VLDL will do the same thing as the kilomicron and as the lipids are released from it, only what's left is, most of it is the cholesterol, and so it becomes the LDL. The LDL will go to the body cells and release some cholesterol, and then that will come back, the remnant of the LDL will come back in the liver to form the HDL, and the HDL will go to the body cells and pick up whatever cholesterol was left behind that was not taken by the body cells, okay, and bring it back to the liver to deal with cholesterol. So diets that are high in trans fatty acids, one of the problems with them is that they can raise the levels of LDL in the body. They are abundant in hydrogenated foods, for example, vegetable oil in the form of margarine, baked goods, and fried foods. This I just told you, where Health Canada requires trans fatty acids content to be listed on the labels. And so to prevent cardiovascular disease, or to reduce it, there are some guidelines that you can follow. So your AMDR for the fat is 20 to 35% of your total calories. Saturated, try to be between 7 and 10%, but if you want to reduce the risks, less than 7% is going to be good for you. Cholesterol, only 200 milligrams per day. Trans fat, reduce to absolute minimum, and increase omega-3 intake, because in Canada, research has shown that we have less omega-3 in our bodies than other places. Okay. Eating smaller meals throughout the day is also good for you, because this doesn't overload the system that I just told you about, right? so you don't have any remaining cholesterol just chilling there, right? Decreasing the amount of food you eat, but eating it more frequently during the day allows for a less saturation of your body in terms of lipid, okay? In terms of cholesterol remnant, for example. Consuming less alcohol in a day, obviously, especially at this age where everyone is like partying and drinking to the point where they cannot even get out of bed, right? So this is important. And actually, drinking too much leads to a disease called fatty liver disease, because your liver will kind of deal with the fat, as I told you. And so if you've saturated with the fat, because alcohols are converted into lipids, then you'll have fatty liver disease, which is basically your liver with a layer of fat in it, and that can cause many problems, including cancer. It's like the last step of liver disease, right? Maintaining an active lifestyle, of course, to lose weight, to make sure that you burn fat. Balancing calorie intake and physical activity. This is important because this is a good way to burn calories, but if you don't change your diet as well, then you end up taking up too much calorie, and this is just useless. Salt intake. So this is unrelated to lipids, really. It's mostly related to the fact that salts increase blood pressure due to the osmosis kind of part of the role that salt play into the fluid balance of your cells. Let's just call it this way. There are medicines that help reduce cardiovascular disease. For example, statins are given to people that have high cholesterol levels, so there are endogenous cholesterol synthesis inhibitors. They will inhibit the pathway that makes cholesterol. This pathway is the mevalonate pathway. There are these bile acid sequestrants like cholecystiramin, which is given to people that have issues with their bile to decrease the chance of having a bile, what do you call it? Not rock, but the thing that blocks your gallbladder, okay? Whatever word. In French, it's caillou, okay? So it basically will carry, jeez, go on Google, it's annoying. Bile, caillou, jeez. Bile stones, jeez, that was very hard. Okay, bile stones. Very complicated to know a few languages. You lose words in one or the other. Nicotinic acid also is given to reduce VLDL synthesis and LDL synthesis and increase the levels of HDL. Nicotinic acid is vitamin B3. So this is interesting because vitamin B3 is important for the synthesis of these two. And so if you remove it, you make less of these and more of this. Okay? So that's pretty much for this lecture. I will be posting this now. The next one should be coming in the next hours, hopefully. Keep studying for the exam until then.